Welcome back, folks. This is my video lecture on a 3-point genetics cross, primarily to map genes along a single chromosome. I find that it's always convenient to first go through a quick review of what happens when we have Mendelian inheritance, where the three genes that we're going to be talking about, J, K, and L, are not on the same chromosome. So that sets the stage. So let's get started with that. Assume that we're looking at a diploid organism, a salmon, for instance. Salmon can spawn and have lots of babies. So assume that our salmon, she's going to have 1,000 babies, right? And we can then quantify the types of babies that are born according to their phenotype. So let's start with that. So we have a fish, and she's going to have 1,000 offspring. And we're going to be able to genotype those offspring. To set the stage, let's assume that we're looking at three genes in this fish. Gene J, that comes in two alternative forms, big J and small j. Gene K, which also comes in two forms, two alleles, big K and small k. And gene L, that similarly comes in two alternative alleles. And we're going to follow these three genes into the offspring that she's producing, these 1,000 babies. So let's look inside the mother, right? This is where the magic happens. And suppose that she has a large chromosome here, just because she's diploid. And then she has a medium pair of chromosomes. And then she has a small pair of chromosomes. And let's assume for this first slide that each of those genes is on one of those three chromosomes. So let's put J in its heterozygous form on the first chromosome. Let's put K in its heterozygous form on the second chromosome. And let's put L in its heterozygous form on the last chromosome. So when these cells undergo oogenesis, i.e. the production of eggs, they have to go through meiosis. And as you know, in meiosis 1, the chromosomes, they line up in the middle of the cell. So if this is the cell here, the chromosomes will line up, and then the chromosomes will be pulled to opposite parts of the cell by the spindle apparatus. Now, if you notice, I did something, I put the capital letters on top in each case. So the chromosome carrying the dominant allele was always on top. But as you know, this is not the case. It's a random process. So sometimes big J will be sitting on top and small j will be underneath. But 50% of the time, it'll be the other way around, with big J on the bottom and small j on top. Likewise, the second chromosome also independently can line up in either direction, with big K either on top or on bottom. And that is a random process. Again, about 50% of the time it'll be on top and 50% of the time on the bottom. And likewise, for the small tiny chromosome on the end, the L, the big L, will be on top 50% of the time, and in 50% of the other cases of meiosis, it'll be on the bottom. So if that's the case, we can get from a single gene with two alleles, we can get two types of egg. Those eggs that contain big J, and those eggs that can contain small j. Okay? But the eggs, in order to be viable, they must carry the middle chromosome and the small chromosome. So they have to carry a copy of the middle chromosome and the little chromosome, like that. So the question then becomes, what do they get with respect to the middle chromosome? Do they get a big K or do they get a small k? And the answer is, it's 50-50. So sometimes the first egg that you see here will get a big K. Sometimes it'll get a small k. And likewise with the tiny little chromosome on the end, right? Sometimes it'll get a small L, sometimes it'll get a big L. And if you look at all these permutations, you can end up with, well, the formula is the number of alleles, which is 2, times the number of genes, in this case, is 3. So we have 3 genes, 1, 2, 3, and that goes there. And because we have 2 alleles for each gene, that number goes in there. So that equals 2 times 2 times 2. And 2 times 2 is 4, times 2 is 8. So we're looking for 8 types of eggs that she can produce, with those chromosomes randomly going into the different types of gametes, her eggs. Okay, so let's tidy this up a bit on the next slide. She could produce 1, 2, 3, 4, 5, 6, 7, 8 types of egg, right? So egg types. And what is going to be the genotype of each egg? Well, sometimes the chromosome will be big J. So we can put that down here. And sometimes the chromosome will be small j. So let's say that half the eggs, the top 4, are big J. Let's keep things nice and simple. And the bottom 4 are small j. Like that. So that's your big chromosome out of the way, right? What about the medium-sized chromosome? Well, the medium-sized chromosome contained the K gene. And in half the eggs, they will get big K, and the other half will get small k in a random fashion. So let's assume that this one gets big K, this one gets big K, okay? And this one gets small k, this one gets small k, and this one gets big K, this one gets big K, this one gets small k, this one gets small k, right? So we'll put underline on the small k's so we don't get confused between the capital and the lowercase rendition of that letter. And then likewise, we have, out of the 8 types of egg that she can produce, we have the baby chromosome, the tiny little chromosome, and that carries the L gene. And again, that lines up randomly compared to the other 2 chromosomes. So in some cases, we'll have big L, and in other cases, we'll have small L. So let's do that all the way down. Now, if you look very carefully, none of those 8 types of genotype are exactly the same, okay? So if you look carefully, none of them will be the same. So let's go for number 4. So in the case of number 4, we have big J, small k, big L. Look at number 6 randomly, all right? We have small j, big K, small l, number 6. So those are the 8 types of gametes that we predicted that she could make if she has 3 genes with 2 alleles each, okay? So that's very important, because when we do the 3-factor cross in the next few moments, we're going to be dealing with 3 genes with 2 alleles each. So now you will know why we're expecting to get 8 classes of egg, okay? Now, in a 3-factor cross, we will take these eggs from this female, who's triple heterozygous. She's heterozygous for J, she's heterozygous for K, as you can see here. She's heterozygous for J, she's heterozygous for K, and she's heterozygous for L. We're going to breed her with a male, so the sperm from the male salmon, we control that. We make sure that all he has for the 3 genes is lowercase, lowercase, lowercase. So homozygous recessive, homozygous recessive, homozygous recessive. So in his case, he has the chromosomes small j, that pair there, the large pair. Then for the middle-sized pair, he has small k and small k. And for the baby chromosomes, he has small l and small l, right? So he was bred to be a tester, a triple homozygous recessive. And that's important too in a 3-point cross, is that one of the parents has to be homozygous recessive, or at least called a tester. Now, his sperm will all be the same because there's no choice, right? So his sperm, in all cases, will be for the first chromosome, small j, for the middle chromosome, small k, and for the baby chromosome, small l. So when those 8 eggs from the mother salmon that we just calculated are fertilized with those sperm, then you will see that the male really doesn't contribute anything other than small j, small k, and small l. Then if you assume that the big J makes the salmon large versus small j makes it small, and the second gene K makes it red and the small k makes it white, and for instance, the third gene L gives it large eyes versus small l, which gives it small eyes, then you can see the egg will determine the actual phenotype of the babies, the 1,000 babies that are born. Okay, so in this case, if this sperm was to fertilize number 7, then the baby there would be such. It would be small j, right? Because small j from the father and small j from the mother. It would be small k and big L and small l. So big L came from the mother and small l came from the father. So this will be a small salmon because J determines size. It will be white because K determines color. And L determines eye size. So it'll have large eyes, not small eyes. And you can see the other 7 categories will give you all permutations of those 3 characteristics. And that's important to know. So this is what happens under Mendelian conditions, where we assume that all 3 genes have their own separate chromosomes. Therefore, one is not interfering with the other. And that's the foundation for moving forward with the 3-point cross. So we're going to move over and look at the 3-point cross. We'll consider the salmon again. We'll consider the same fish, right? And we'll consider the same number of babies born, 1,000. But this time, we'll pick a different set of genes that we know are on the same chromosome. So let's go over here to a fresh piece of parchment and put down that scenario, right? So we're going to use a key, and let's use different letters now. Let's use X, Y, and Z, right? So these are now our 3 genes. Again, we're going to make sure that the mother salmon is heterozygous for each of the 3 genes, right? And this time, we're going to assume that if this purple thing is the chromosome, she has 2 chromosomes. And on one chromosome, she has 3 genes close to each other, but we don't know how close they are. And that's the purpose of a 3-point cross, to figure out the distance between X, Y, and Z on the same piece of DNA. But because she's diploid, she'll have a second chromosome. And the second chromosome will carry the 3 same genes, X, Y, and Z, and the alternative alleles. And during meiosis, these 2 chromosomes will come together, and then they'll swap parts, as meiosis wants to encourage during crossing over. And because these chromosomes are coming together, the father's chromosome and the mother's chromosome of this salmon, they will then perform a crossing over. Which gene is in the middle? And how far apart are the genes from each other along the chromosomes, right? Is the distance between the first gene and the second gene bigger than the distance between the second gene and the third gene? A 3-factor cross will answer all those questions for us. And that is what we call a genetic map, all right? So keep that in mind now. We're not going to look at 3 separate pairs of chromosomes. We're looking at one pair of chromosomes with 3 genes along their length. So let's write that down here, right? So we'll start with the same situation. We'll have 1 chromosome now. So 1 bar going right across means 1 chromosome. And we'll have the homologous chromosome like this. And we'll assume that in this case, she is heterozygous for each gene, right? So X, Y, and Z. So that's one configuration that she can be, right? So in this particular case, this salmon that we have, she could have all three dominant alleles, big X, big Y, and big Z, on the top chromosome. But we don't know. And that's what we're trying to find out. So alternative types of salmon could exist. You could have a salmon that has the following situation, right? Where we could have, for instance, we could have the X gene in the middle, right? Like that. Again, we're trying to figure out which one of these two is correct. And the genetic analysis will allow us to work backwards to figure out which of these alternatives is correct. But you might be guessing there's a third alternative too, in that we could actually have the following situation where Z is in the middle and X and Y are on the sides. Okay, so in this case, we could have the following genetic organization. I'm going to get the X's, the Y's, and the Z's together so I don't make a mistake. All right, and the data will allow us to answer that question. Which gene is in the middle? But we also have a second big question. How far apart are the genes from each other once we determine which one's in the middle? And a 3-factor genetic cross will allow us to work all this out. What we're going to do is deal with all three situations simultaneously. So you can see how the data changes depending on which gene is in the middle. So let's divide the board into three. Let's use a different color to divide. So here we have one division, two division, and then of course we have the third division, right? So for each one, we need to generate our 8 classes of egg, right? 8 classes of egg, just like that. 8 classes of egg that we have here. We need to now do the same thing with our linked genes on the same chromosome. So let's put that here. 1, 2, 3, 4, 5, 6, 7, and 8. Let's start with example A. So in example A, we're going to assume that meiosis is taking place, right? And this is happening inside a cell. Assume that for this pair of chromosomes, that there is no crossing over. No crossing over, right? So for category 1 and for category 2, we're going to assume that the chromosomes are going into the eggs unchanged. So here, for number 1, we have the top chromosome. And that's going to give us X, Y, Z, all capitals, right? And that's going to be the first type of egg. And then the bottom chromosome will move into its own egg because it hasn't exchanged any parts with the top chromosome as it is, right? So that's going to be number 2. So that's going to be small x, small y, small z, all right? So there's your parental type of chromosomes. Now they're called parentals because there's no crossing over. They're the same configuration as in the mother, right? So the eggs get exactly the same chromosomes that the mother had, and they just get segregated into 2 gametes, 2 eggs. Likewise, for salmon B, we get the same 2 chromosomes unaltered going down into the eggs. So in this case, for category 1, we will have Y, capital, X, and Z. So that would be one type of egg. And then we'll have the bottom chromosome going in unaltered into its own eggs as small y, small x, and z, small z, right? Okay, we have the top chromosome going in unaltered, but the genes are assumed to be in that order there. So we have the 3 orders, and we're going to have to figure out which one of those 3 is correct, A, B, or C, right? And then let's finish off here. So the second major type of egg will be small y, small z, and small x. And all of them will be parental types, right? Because they represent the red ones just moving directly into the gametes without any crossing over. All right, so far so good. Let's move on and use a different pencil to illustrate something else. Now let's assume that we get crossover. Let's use that color. Say we get crossover here between the first gene and the second gene in each case, right? So the first gene and the second gene. So we're getting crossover. This is a product of meiosis. Meiosis 1 is you get crossover between the homologous chromosomes, right? And assume that there is crossover and it happens between the first and the second gene in each of these three scenarios, right? So when the crossover happens, you get the following condition. Let's put that in yellow. So you get this chromosome crossing over and going down there. So now that egg will get that fragment of chromosome. So let's put that down in our analysis. Let's use blue now for that. So number 3, we will have big X there, then moving down to small y and small z. And that's going to be an alternative type of egg that contains that chromosome. Now the reciprocal of that is the other way around. So let's use a different color to illustrate that. So what we're saying here is that the other chromosome will be that and that. And so we need to record that too. So the fourth type of chromosome that's possible from this female salmon is the following. So we start with small x, and then we move from the bottom chromosome, as the green indicates, to the top chromosome. And we end up with this. So that's going to be recombination between the first and the second, between the first and the second gene on that chromosome, right? And same thing here. So the first gene, second gene, and the third gene, right? First gene, second gene, third gene. First gene, second gene, third gene. So we keep the order of the genes left to right. Let's go straight away and do B, because she has the same thing, right? So 3 and 4 can be put down as reciprocal. So we start on the top here, and we come downstairs. So it's going to be big Y, and then small x, and then small z. Okay, that's a Z, by the way. And then we have the bottom chromosome going upstairs after it crosses over. So there's going to be small y, big X, big Z. So again, that's going to be the same. Crossover between the first gene and the second gene in B. Let's do the same thing with C. So here we have crossover right there. And we start with the top chromosome coming down and crossing over to the bottom chromosome. So there's going to be big Y, small z, and small x. Okay, it's going to be one chromosome, underscore. And then the other chromosome will go into the other egg, and we'll get the opposite. So start at the bottom now, and then move upstairs. So small y, z, and big X. There you go. Okay, so all that is the same thing. So let's change our direction a little bit. Let's redraw things, and let's look at a different scenario now. Now we're going to look at crossing over between the second gene and the third gene, right? So these red dots that I'm putting in, they will represent the single crossover between the second gene and the third gene. So now we're no longer looking at the crossover between the first gene and the second. So what we need to do in order to keep things nice and tidy is maybe I need to redraw this, right? So that we don't get confused. Okay, so let's redraw that and keep it nice and tidy. So what we had was we had X, Y, and Z on top, and we had the lowercase x, y, and z on the bottom. And now we're going to have a crossover right there between the second and third gene, right? So between the second and third gene, between the second and third gene. So when you get crossover there, you get the following condition. So you get the top chromosome then crossing over to the bottom chromosome, right? So we need to now translate that, and let's use a different color pen. Let's use orange for number 5 and 6. So now we have big X, big Y, and small z as one chromosome. And then the reciprocal of that is small x, small y, and then big Z. Okay, so that's starting at the bottom here and then moving upstairs. Let's do the same thing with B. So we have 5 and 6. So we start on top, then we move downstairs like that. So Y, big X, and small z. All right, that's going to be one chromosome. And then the reciprocal of that will be the other one here. That's going to be small y, small x, and big Z. Okay, let's move over to C. And with C, the same thing. We have the top chromosome crossing down to the bottom chromosome with that crossover between gene 2 and gene 3. So for 5, we have big Y, big Z, and small x. Right? And then for 6, we have the opposite. We start at the bottom, and then we cross over to the top between genes 2 and 3. There we go, right? So we have 2 more types of egg to produce for each of these possibilities. And that's going to be 7 and 8, right? Now, what could happen? Well, there is the possibility, even though rare, there's still a possibility that you could get a crossover between gene 1 and 2 and another crossover between gene 2 and 3. So this is your double crossover, right? So let's write that down here. So this is going to be a crossover between 1 and 2 and another crossover between 2 and 3. Okay? So crossover between 1 and 2 and crossover again between 2 and 3. So how does that work in our figure? Well, let's go back and redraw the first one. And let's get rid of everything else. Okay, so let's redraw that one to save time. We won't draw the others. So we had X, Y, Z on one chromosome. Okay, so that would be one chromosome, X, Y, Z. And on the bottom chromosome, we have X, Y, and Z. So let's put that down. But this time there'll be lowercase X, Y, and Z. And what we're saying this time is that we get 2 crossovers. We get a crossover here and a crossover there. Very unlikely, but it's possible. And it does happen when you have a large number of offspring. So here, let's use a yellow to start at the top. So you got this, 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 and then back to the top again, right? Because 2 crossovers, one next to the other, will turn the chromosome twice. So once you move downstairs, then you move upstairs again, right? So that chromosome has to now be written on the page. Let's use a different color. Let's use green for 7 and 8. Okay, so 7 will be starting on top, moving down to the bottom of the second gene, and then moving back upstairs to the last gene. There you go. And then we have the reciprocal. Starting at the bottom, we have small x, then we move upstairs to the big Y, and then we come back downstairs to the small z. So those are the 8 types of gametes that she can produce if the Y was in the middle. If you could continue now with B, if that was the case, then you would have the following, right? So you get double crossover here, and you get that. So that would be big Y, small x, and big Z. And then the last one down the bottom would be small y, big X, and then small z. Okay. And then for example C, if that was the case, if the Z gene was in the middle, then you would get this. So you start here, come downstairs, and then go back upstairs again, right? So big Y, small z, and big X. And then the bottom starting chromosome is small y, big Z, and then small x. And those represent the double crossovers. So here's the gist of all this, right? Here's the kicker. Of all these 8 types of gametes for A, which ones would be the most likely to exist in nature, assuming that crossover is rare? That crossing over between chromosomes is infrequent. So think about that. Which one of these 8 classes here will be the most common? Well, the answer is those that do not have any crossovers. And those will be the parental types, right? These two. These two would be the most common. So suppose you had 2,000 babies, right? How many babies of that type would be born? Well, that depends on how far the genes are on the chromosomes. But let's just give you some hypothetical so you can put that number into your head, right? So let's get rid of these check marks for now. And let's put numbers in its place. So suppose this salmon produced 2,000 gametes. Let's put 2,000 here. Of the 2,000, some types will be more common than others. So say out of the 2,000, there were 500 of this type and 500 eggs of that type. So that's 1,000 already. But those are the most common because they have no crossover. Then, of the remaining 6 types below, the blue type, the orange type, and the green type, which types will be the next most common type of egg that you find in the population, right, of salmon? Well, those will be the ones that have a single crossover. So those will be 3, 4, 5, and 6. So let's assume that there's 200 of each of those. And the actual numbers can vary depending on how far the actual genes are from each other, right? But the last thing I want you to understand is which types of gametes, which types of eggs will be the rarest. And of course, those will be the ones that have 2 crossovers, the double crossovers. And the only ones that have double crossovers are 7 and 8, right? And in our example, maybe you'll just have 100 of each, or maybe even less, right? So you can see the numbers are not what you expected based on here. In this case, because we have 3 separate chromosomes, each chromosome, each type will be 250. So we expect to find 250 eggs of each type because each chromosome is independently segregating inside that particular condition, right? So J, K, and L are all independent of each other, then each class is available in equal quantities. And that's what you expect to see. But if you don't see those numbers, when you look at the babies, you see something strange, such as this, then you know that the genes are on the same chromosome and they're close together. Now, the data in A will be produced if Y was in the middle. If X happened to be in the middle, because you don't know this, because that's what you're trying to determine, then the same numbers will be over here. That'll be 500. This will be 500. So you can see that in this case, the wild types are both the same. The wild type chromosomes, the parental chromosomes, are the same, right? But later on, you'll see that they become different. And same thing here, right? You get 200 of these, 200 of these, 200 of these, 200 of these, 100 of those, and 100 of those. If the genes were the same distance apart, but the order was that X was in the middle, right? So here we have Y in the middle, here we have X in the middle, and here we have Z in the middle. And here you would then see 500 of these, 500 of those, 200 of those, 200 of those, 200 of those, and then just 100 of the bottom ones. So that's a departure from these numbers. And that just tells you that these 3 genes are now linked on the same physical piece of chromosome. But how can you use these numbers to tell you the order. So on the last slide, which I need to show you in a second, I'm going to show you actual data. And then what you have to determine is which gene is in the middle. Is it A, is it B, or is it C? And the numbers will tell you, because the most frequently produced babies that come from the first two types of parental chromosomes, these types, will be the most common. And then these will be the least common babies born to this salmon. I took the liberty of putting some data together for us on this particular slide. Your job is to give us three answers. The first answer is which allele is on which chromosome. Now, students assume that big Z, big X, and big Y are all on the same chromosome, and that the other chromosome, the other homologous chromosome, it carries the small X, the small X, and the small Y. But why? You don't know that. You're assuming that. So if you assume things, you get things wrong, right? So what we're looking at is what did the grandparents of our fish give it, right? And there could have been recombination in the grandparents that mixed up the genes so that the alleles were on different chromosomes. So don't assume that. So the data itself, this data, is going to answer that question for you. So let's go ahead and answer question number one, right? What did the chromosomes of the mother fish look like? And that's easy to find. All you have to do is identify the two largest categories of babies. And if you look down here, we can easily see that. Let me change my pen to a green pen. You can see that this number represents one of the largest classes of babies. And then looking down the list, number 6 represents also the largest class. So those two dots are now indicating the genotype of the chromosome, which we call the haplotype, right? They represent the chromosomes that were actually present inside the eggs from which these babies came. And they represent the most likely event, which is no crossover. So crossovers are rare. Remember that, right? Just like a meteor hitting the earth, crossovers are rare. So the most normal condition is no crossover. And looking over to the right, you can see we have two categories of no crossover. And those are the parental chromosomes, unaltered, haven't gone through recombination, haven't gone through crossing over, right? And they appear as they are. So let's move those chromosomes, the ones with the dots, over to the far side, right? So we're going to take this chromosome here and put it on the far side, because that is how it came unaltered from the mother. And then we're going to move this one too to the far side, because that represents the second chromosome that came unaltered from the mother. You can already see, right? It doesn't look like any pattern that we looked at on the previous slide. So here we assumed that all the chromosomes carried either the dominant combination or the recessive combination of genes. But that's a false assumption, right? So the data is telling us exactly what the chromosomes look like with respect to these three genes and their different alleles. So there is your first answer. What did the chromosomes look like? Well, one chromosome had big X, big Y, and small Z allele. And the other chromosome in the mother had small x, small y, and big Z on its structure. So this one had big X, big Y, and small z, and this one had small x, small y, and big Z, okay? So that's your first answer. Now, in a very quick step, we're going to answer the second question of the three. Which gene of those three, X, Y, and Z, is in the middle? Now, you may assume, again, that Y is in the middle because we wrote it in the middle. But that's just a placeholder. That's just for convenience. The actual data will reveal to us the secret, i.e., which gene sits in the middle. Now, let's think about it. If you take the parental chromosomes and you perform the rarest event, which is a double crossover, then those numbers will represent the smallest number of offspring. So go back to your data set, and now let's use a different color to highlight two different numbers, the lowest numbers now. So we looked at the highest numbers. They tell us the haplotype of the chromosomes. Haplotype simply means what each chromosome physically looks like, and we just said that. Now look at the lowest numbers, and number 4 and number 7, they give us the lowest numbers. Out of the 2,000, 20 babies of type 4 and 10 babies of type 7. So let's move those chromosomes over to the far side and put them next to the double crossover. And the double crossover is, of course, 7 and 8. So let's move those chromosomes over here. So let's take this one here and move it down here, right? So that's going to be big X, small y, and big Z, all right? And then this one here, let's move that across, small x, big Y, small z. All right. So what we have to do now, and this is the secret sauce, we have to take the parental chromosomes here and perform a double crossover and get to these results. If we can't get to those results with a double crossover between the first gene, second gene, and the third gene, then the order of the chromosomes that we wrote down for our convenience is not the real order in nature, right? So let's assume that we have what we're going to do for convenience, we're going to move the parental chromosomes to the middle of the whiteboard so that we can calculate what the outcome of a double crossover would be. So let's take this chromosome here and move it right into the middle as it is. And let's put underneath that the second chromosome in the parent. Now what we have to show is that when we get a double crossover, like that, we can actually produce one of these two. And if we cannot produce one of those two with that double crossover, then we assumed wrong. We put the Y in the middle, then that's not the case. So let's have a look at this, all right? So let's follow the lines. Let's use a different color. Let's follow the lines. So big X, small y, small z, right? So that's what it's saying, that with a double crossover, that's what you should get, right? So it's saying big X, small y, small z. So do either one of these look like that? So big X, so we have big X here. Small y, so far so good. Oh, no, sorry, it fails. It failed, right? Because it failed, we assumed that Y was in the middle, Y is not in the middle. Let's confirm that by looking at the reciprocal chromosome. So the reciprocal chromosome in this case will be this one here. Let's check that one out too, right? So if that's the case, then we should be getting, from that one, we should be getting X, then moving over to the top, and then coming back down to the bottom, right? So do either one of these look like that, 7 and 8? Let's have a look. So small x, big Y, big Z. Small x, big Y, big Z. Sorry, it failed on that too. So that cannot be the order of the chromosomes. So even though we wrote them down, X, Y, Z, that's just the way that humans think, right? So what we have to do now is abandon that and then reorganize the 3 genes. Let's try putting Z in the middle, right? Let's try putting Z in the middle. So let's try that. So let's rewrite this by putting Z in the middle. Let's get rid of all this. Okay, so what I'm going to do in my head, and actually on the whiteboard, is I'm going to move this chromosome down here, but at the same time, I'm going to move Z into the middle, right? So now we're going to try putting Z in the middle. So it's going to be X, Z, Y, right? That's the top chromosome, right? That's the Z. And then the bottom chromosome, again, you're going to put Z in the middle, right? Like that. So let's move that across. That's going to be X, Z, Y, right? So I've just written it down again, but this time I moved Z into the middle. Now we're going to look at the data again and see if we can explain that. So let's see if we can do that. So we should be able to produce from that data set a chromosome that looks like this. So big X, starting here, coming down there and back up again. Big Z, big Y. So do either one of these two, 7 and 8, look like that? No, neither one does. So our assumption that Z is in the middle is also wrong. Also wrong, because we're not seeing it in 7 and 8. We have one more possibility left. Let's put X in the middle this time, right? We haven't had X in the middle. So let's try that again. So let's erase this. And let's put X in the middle, right? So just copy it down here again. So this time I want to mentally put X in the middle and then draw that accordingly. So here's my chromosomes. So let's put the top one in the middle. So it's going to be Y, X in the middle, Z, right? And now let's draw the bottom one, Y, X, and Z. Now we're going to see if a double crossover can produce either one of 7 or 8. So let's follow that through and let's see what we get. So we have big Y, small x, small z. So big Y, small x, small z. Okay, and the bottom one, the bottom chromosome, that would give us this crossover, right? Double crossover. And that could be written like this. So it's going to be small y, big X, and then big Z. So let's move our attention down to 7 and 8 again here. Do either one of them, well, not either one, do both of them resemble both those chromosomes above? So let's check this one, right? So we have wrong slide. We have small x with big Y with small z. We have small x with big Y, small z. Hallelujah, right? If you don't mind me saying that. And then let's check the other one, this one. And that should be the one on top at number 7, right? So big X, small y, big Z. Big X, small y, big Z. And once again. Now we have confirmed number 2, which gene was in the middle. So this data tells you the haplotype of the parental chromosomes, what the mother had before she went to produce her eggs. And then this data here tells you which gene is in the middle. So we've satisfied 2 of the 3 things that we're after. The final thing is to work out the distance between the 3 genes. Now that we know that X is in the middle, yes, we want to know how far is Y from X and how far is Z from X. We need to know the distance. And guess what? This data here that we have on this slide will tell us, right? How will it tell us? Because we can use the panel on the right-hand side, this panel here, to help us. Okay, so we're going to do that right now. So the order of the genes is going to be maybe Y, X, Z. Now it could have been Z, X, Y. It really doesn't matter. We just don't care about the 2 on each side. We want to know which one was in the middle. And we just found that out by looking at categories 7 and 8. So the other data sets that we haven't dotted yet, the other 4, 2, 3, 5, and 8, they're going to help us calculate the distance between X and the 2 outside genes. So now that we have found out which gene is in the middle, the X, let's rewrite these genotypes according to that. So we said X is in the middle with Y on one side and Z on the other, okay? So it doesn't really matter which side Z is on or Y. All that matters is that X is in the middle. So let's rewrite this in that new format, okay? So we have Y, X, and Z. All right, so that's the new order. So we can get rid of these. So what we're going to do now is the last part of the exercise, which is to work out the distance between these 2 genes, the middle X and the gene that we put on the left-hand side. So if the original configuration is like this, we can see, if we change the color slightly, that either the small x is with the small y, or the big X is big with the big Y in the parental chromosome. So anytime that we have a recombination of that, we want to make a note of that. So let's use a different color pencil. Let's use a purple felt tip. So if that's the parental type, now you can see here, we have a big Y with a small x, which is different to what we had earlier, right? And so what we do is we just go up and down looking for those changes. Here's another change, here's another change, and here's another change, okay? So those 4 out of the 8 represent a rearrangement between big Y and big X, or between small y and small x. So those are the recombinants. So we add them up. Yes, we add up those numbers. Let's do it in black. So we say 150, because that's the number over here for this purple. Then this number over here for this purple, plus 20, plus 140 for this purple, plus 10 for that purple. Okay, so we add them up and then we divide by the total, which is 2,000. Why? Because we had 2,000 over here. So 150 plus 20 plus 140 plus 10, I believe is 320 divided by 2,000. And then you multiply that by 100 to get your percentage, right? So that's going to be 0.16, and then 0.16 times 100 equals 16. So there's going to be 16% or 16 map units, right? Either way. So the distance between X and Y is 16 map units. Now we're going to work out the distance between X and Z. And the way you do that, you go back to your data, and now you're going to identify any regions where big X with small z, or you have here small x with big Z. So any departure from that, you're going to highlight, and then you're going to add up those numbers as recombinants, right? So those are the parental types. So let's have a look around. So let's have a look at the first data set. So right here, we have a different combination. So we're going to include this data. Right there, we're going to include that data. Then right here, we're going to include that data. And then right there, we're going to include that data. Okay? So the data with the green dots, we're going to include. So let's put that in our formula. So first one we have here is 330. Okay? And then the next one we have is this one. So we're going to include 20. And then the next one we have is this one. We're going to include 10. And then finally, we should have one more. There it is. And we're going to include 350. So we know that X is in the middle with Y on one side and Z on the other. And the distance between X and Y is 16 map units. And the distance between X and Z is 35.5 map units. So those lie on the same chromosome at that. So 16 map units is less, so X is closer to Y than it is to Z. And that's how you solve a 3-factor cross. Thank you.